Assalamualaikum and hi, my name is Wan Nur Sharafana Alimi Tuan Hassan. I'm a degree student from University Teknologi Mara, Cawangan Negeri 9 Kampus Kuala Pilah. So I would like to share about my research project entitled Molecular Identification of La Gibbon, Halobits La in Captivity using Mitochondrial DNA of Cyt-B region.
The white-handed gibbon, scientifically known as Halobits la or other name is La gibbon and ungka tangan putih, belongs to the family Hylobatidae. This species is native to Southeast Asia and is also widely distributed across peninsular Malaysia. So this gibbon characterized by having long arms, hooked hands, small size with dense fur, hairless face, and also white hands and feet.
So, the problem statement of this research project is a traditional morphological identification of La gibbons often leads to misidentification, which can hinder conservation efforts, affect the relocation programs, and complicate breeding and reintroduction decisions. So, for this project, there are two objectives. The first one is to optimize the PCR conditions for Cyt-B region amplification, and the second one is to identify captive white-handed gibbons using mtDNA of Cyt-B region. So in order to complete these research projects, here are the flow of the methodology, starting from the fecal sampling, and then proceed with the genomic DNA extraction, and then proceed with the gel electrophoresis to identify the presence of DNA from the fecal sample, and then DNA amplification by the PCR to amplify the DNA fragment, and then proceed with the gel electrophoresis, and then lastly will be end with the DNA sequencing. The sample collection was made at the Zoo Melaka, so there are 8 total samples were collected, 4 from the island and 4 from the zoo clinic. So the sample collection was helped by the zoo staff and as well as my SV and my partner of this FYP project. So next is the genomic DNA extraction. So all the samples were extracted using the analytic GENA kit and then labeled and stored at the negative 20 degrees Celsius in a final elution of 100 microliter. So the third step is gel electrophoresis after the genomic DNA extraction. So as you can see on the left side, 4 samples from the zoo clinic were successfully extracted and visualized under the UV light. The gel showed DNA separation, confirming successful extraction and also purification, but the bands appeared faint and also smeared. This may due to the DNA degradation, low quantity or inhibitors, which are common challenges in non-invasive sampling, which is fecal sampling. So next is PCR amplification. So during this research project, the PCR optimization also conducted in order to know the optimum temperature for annealing, and also the PCR optimization was proceeded by using this cocktail, and then the forward primer and reverse primers as follows. So for the PCR optimization, we conduct the gradient PCR in terms of the cycles. From 30 cycles, we increase to 40 cycles, and also in terms of the annealing temperature, we range from the 43.5 degrees Celsius until 52 degrees Celsius. So after the PCR optimization, we proceed with the gel electrophoresis for the second time. So as you can see on the left side, 775 BP region of the Cyt-B gene was successfully amplified via the PCR using the species-specific primers after optimizing PCR parameters. So the gel electrophoresis showed the brightest band in lane 4 at 48.8 degrees Celsius, indicating the optimal annealing temperature. So the PCR optimization is crucial for successful results, especially in ancient DNA studies. So this is the last step, which is DNA sequencing. So the PCR product will be sent to the Apical Scientific Sendirian Berhad for sequencing. So after we receive the sequence from them, we will blast at the NCBI blast, which is under the nucleotide blast. So this is the result that we will get after we blast the sequence at NCBI blast. The Cyt-B region, approximate 700 base pair, was successfully sequenced for 1 out of 4 samples. The sequence matched a known gene bank entry with 98% query coverage, 99.47% identity, and an e-value 0.0, confirming its significance. So this sample was identified as Halobitsla. As a conclusion, the study successfully achieved its objectives by optimizing PCR conditions and accurately identifying the species. Troubleshooting, gradient PCR, and cycle adjustment lead to the successful amplification for sequencing. The Cyt-B region proved to be a reliable marker for identifying the white-handed gibbons. So before we end, I would like to thank Encik Rafiq and Dr. Zahidah from Zoom Laka for their invaluable help in sample collection, and special gratitude goes to Dr. Nur Azimah binti Osman for guiding and support. So this study was made possible with a research permit from the Department of Wildlife and National Parks Peninsular Malaysia.